A new baby enters the world. And just like every child before him, he carries the expectation of a long and healthy life. But today, that expectation is higher than ever before, because this baby enters a new world, where our greatest dream, to conquer disease and prolong life, may finally come true, through the power of genetic science and DNA. At the start of the 21st century, we are in the middle of a genetic revolution. Driving it is an incredible discovery that through all of us runs a single code of life called DNA. We now know that DNA guides our existence from the moment of our birth to the instant of our death. That our fate is written in this simple molecule and all its thousands of combinations that are our genes. The promise that we would all live longer, healthier, disease-free lives has come with the breaking of that code, first achieved by Watson and Crick at Cambridge University in 1953.
We've been tracking two technology revolutions for the last 40 years, computers and genes. In the last several years, these defining technologies of the 21st century have begun to fuse together to create a powerful moment for a new economic and social paradigm in the 21st century. Genes are the raw resource of the 21st century.
The question is whether we're on the verge of conquering most of our medical problems, our major diseases, things that makes us go wrong. And the answer is absolutely yes.
We're investing heavily in our genetic future. It's estimated that there are more than 1,500 labs working on genetic medicine in the US alone.
Our technology is going to have enormous influences on who we are in the future. Essentially, we have slammed evolution into fast forward.
But where some are happy to embrace an unknown future, others see too much faith placed in untried technology.
I think to believe that the scientific community has the wisdom and the knowledge and the foresight to create a second genesis that can perfect the first genesis, millions of years of evolution, is absolutely, it's pathological thinking.
But what do ordinary people think? To find out, Discovery conducted a comprehensive poll to sample public opinion on genetic science in 8 countries around the world. First of all, we asked if genetic science would benefit mankind. 2/3 of Britons believe it will be beneficial, with the Americans feeling more positive at 82%.
It opens up this whole new sort of realm of possibility, which can be really positive, I think.
I'm hopeful for the future with DNA because it's going to help eradicate disease.
Oh no, I think it's very exciting. I think science has got to move forward.
But many people believe that human genetic research is tampering with nature and as such is potentially dangerous. Most concerned are the British and the Poles. Least worried are the Danes.
I don't think it's going to help anybody in as much as what have they done with it?
It's very frightening overall to me because it's basically humans playing God and creating new life form, and it's just not, in my opinion, what we're meant to do with nature.
It's just not. It's not natural and it's not God's way.
On balance, when asked if the benefits from genetic developments will outweigh the risks, just over half agreed, though the British were the least convinced. But whatever people believe the future may hold, the fact is that the humble DNA molecule is affecting real people now. Genetic science is already transforming lives. Disease prevention has long been the dream of medicine, and genetic science could make that dream come true. For every child born at the start of this century, the hope is that by the time he has grown, we will be able to read every part of his genetic makeup and determine his risks from all major diseases. And forewarned is forearmed. because it's now realized that the great killers of the Western world, like heart disease, diabetes and cancer, all have a genetic basis. Together, they account for half of all adult deaths in the UK. One of the biggest killers among women is breast cancer. Every year, 38,000 women find out the hard way that they are at risk. Wendy Watson was one of them.
My mother died when I was 16. She died from breast cancer. She was 44. Her mother had had breast cancer in both breasts and died from ovarian cancer. So I went to the GP after my mother died and said, could it possibly be hereditary? And he said, oh no, don't be so silly, it's breast cancer isn't hereditary.
But when Wendy researched her family history, she found overwhelming evidence that the cancer was passed from generation to generation. 9 out of 10 of her female relatives had developed breast cancer.
And I thought, I don't actually want to spend the rest of my life worrying about when I'm going to get it. And I came to the conclusion that the best thing I could do would be to remove the risk as soon as possible, i.e. remove it now, before I've got it.
As the cancer starts in the breasts, the only sure way of preventing it from spreading is to have the breast removed. At the age of 35, Wendy Watson underwent a preemptive double mastectomy.
What I did isn't right for everybody, but it was absolutely right for me. Absolutely. And I went into this having opened my own eyes and looked into it for myself, and I was very comfortable with what I'd done. I was supposed to go back 6 months later and have reconstructive surgery. I didn't because I couldn't decide what size to be. And 10 years on, I haven't got a clue what size to be now, and I can't be bothered either.
Wendy had her operation without knowing whether she had actually inherited breast cancer or not. The evidence, though compelling, was only circumstantial. The promise of genetic science is to take the guesswork out of disease prediction. And this is the hope for Wendy's daughter, Becky. But it depends on finding the genetic cause of breast cancer. Our genetic fate is sealed the moment we are conceived. In that fusion of the egg and sperm, the first cell has 46 chromosomes, 23 from the mother and 23 from the father. Making up each chromosome, wrapped in a tightly packed bundle, is our DNA, the code of life. The DNA code is made of 2 matching strings of chemical bases, joined and twisted into the famous double helix structure. Each of the 4 bases pairs up with a partner on the other strand, A to T and C to G. These bases then arrange themselves into bunches, tens of thousands at a time. These bunches are our genes. It's the pattern of gene activity which makes all the different cells in our body. But what makes us all different from each other is which genes we inherit from our parents. And that is largely a game of chance. When a couple have a child, half the mother's genes and half those of the father are combined to make the new baby. We all hope our kids will be dealt a good hand and inherit a good combination of genes. Every time we conceive a child, the genes get reshuffled into a new and unique combination. The reason our children are like us is because of where the genetic information comes from. But there's no way of knowing quite how each new generation will turn out. For most, it comes down to things like our height, being short or long-sighted, an aptitude for maths and so on. But occasionally, nature makes a mistake. And a child inherits something from its parents that is altogether more serious. The problem is, with tens of thousands of genes in the human body, how do you find the one that's gone wrong? In Salt Lake City, Utah, scientists managed this improbable feat. Their big advantage lay in the surrounding population. Salt Lake City is home to the Mormon Church. The Mormons believe that if you become a Mormon, so can your ancestors. As each ancestral soul has the chance to be baptized after death, the dead must be accurately identified. Every Mormon family has a record of its genealogy here in the Family History Library, the largest of its kind anywhere in the world. The community is one that is traditionally tight-knit and marries within itself, often producing very large families. For the geneticist, this is a potential goldmine of information that can be used to track down genetic disorders. Dr. Donna Shattuck was part of the breast cancer search.
The idea that there's a single gene responsible for hereditary breast and ovarian cancer comes from the fact that there are families in which there's genetic clusters of the disease. So this is an example of a Utah pedigree in which a disease is segregating in multiple generations. And you can see that the grandfather here is affected with, in this case, prostate cancer. So all the affecteds are males here. The black squares are the affected individuals. As you scan this picture, you'll notice that the black squares, the affected individuals, have the green chromosome. And so then we look for the smallest piece of the green chromosome that goes into an affected individual. And that's how we know where to start looking for the gene.
This is how they track down the breast cancer gene. With a short stretch of chromosome to focus on, they compared that piece with the same stretch from the general population. They found a mutation, an area where the DNA was damaged. The affected gene is enormously important as it acts as a tumor suppressor. But the mutation disables it completely, allowing the cancer to grow unchecked. The mutation was named BRCA1, breast cancer gene 1. Seen here is a single error in a gene nearly 8,000 letters long. It's a G, where it should be a T. Finding BRCA1 was a major milestone in predictive medicine, because from this they were able to make the world's first cancer gene test. Myriad process up to 100 BRCA gene tests every day. Each one is potentially saving a life.
The power of the genetic test is not only to say these genes are involved, but to tell the individuals in a family whether they have the specific individual risk. That's the power of the gene test, and those are the people that you give the interventions to. Predictive medicine is exactly that, assessing the risk and taking steps to reduce the risk of future disease for individuals.
BRCA1 is now thought to be a culprit in nearly 10% of all breast cancer cases. Having it raises a person's chances of getting breast cancer from 5 to 85%. For many adult women, the test for BRCA1 is an early warning system. 2 years after her operation, Wendy Watson was offered the chance to find out if she carried BRCA1. She tested positive.
Peak 107, the 50-50 music mix of yesterday and today. This is Sean and Becky at breakfast.
It makes no difference to Wendy now, but it does affect her daughter.
You've missed the news that the calendar is in.
Every single penny of this goes towards the Genesis appeal, which is to help prevent...
Becky knows she has a 50-50 chance of having inherited the BRCA1 gene.
Sponsors and everything helping us out.
I'm only 21 at the moment, and the genetic test has been available to me since I was 18, which I will definitely take. But breast cancer is in our family doesn't strike till about 35, early 40s. So I'm in no big rush to worry about it.
If you do want your calendar, then you know, come down and get some. Get them in bulk.
At the moment, the only preventative measure that's open to me is to have a double mastectomy. But in 10 to 15 years, who knows what they'll have developed. I mean, I have every faith in things that they'll develop something. But if not, then the double mastectomy is definitely an option.
In Discovery's poll, the vast majority of people in all countries would choose to be tested if there was a history of genetic disease in their family.
If I knew that someone in my family was diagnosed with something that could be determined from some kind of genetic testing, and it's something that if treated early or caught early, would help, would benefit the treatment and that sort of thing, I would be 100% for it, 150%.
It seems to me that if you could determine ahead of time that someone in your genetic line, your parents, grandparents, had a propensity for a particular disease, that you should be tested for it if it could be determined, so that you'd know ahead of time and be prepared for whatever the future would hold.
If you could find a cure for it by stopping it in the early stages, I would surely go for that, yeah.
Gene tests seem to be the great hope of predictive medicine. But at the moment, it's only possible to test for single gene disorders like BRCA1. And that simple test took 6 years to produce. It's now realized that most of the major disorders like heart disease, Alzheimer's, most forms of cancer and diabetes involve a complex pattern of genes. As yet, no one knows how many. And no one knows what most of these genes do. Professor Steve Jones thinks we may need a reality check.
There's a nasty sense that really what we've sequenced, what we think of as genes, may be something much more complicated than we thought. Maybe every one isn't just making one protein and doing one job. Maybe every one is doing dozens of different jobs. Each so-called gene may be a black box with dozens of little units hidden inside it, which are cut and spliced and arranged in a way that we don't understand.
By the mid-1990s, scientists realized that understanding complex genetic diseases meant understanding every single gene in the human body. But they had to find them first. Imagine all the DNA in your body stretched out like a road. Over a journey of 1,000 miles, you would pass 50 base letters every inch. And just like the real landscape, much of that DNA landscape would look the same. Featureless, inactive, vast numbers of repeated letters, much of it seemingly doing nothing. It's estimated that only 3% of DNA is part of a functioning gene. Occasionally, there are areas that make something. These are the genes that are actually producing proteins. What the rest do, no one knows. A lot of it appears to be junk left behind from our evolutionary past.
There is a lot of junk in there, undoubtedly. But the thing which I want to emphasize is it's actually quite straightforward to go and sequence, to get the order of letters, junk and meaning, the whole thing. Now that's 3,000 million letters. Sounds quite a lot, but it's not actually that much in terms of computation. With a little compression, you can write it on a single CD-ROM. You can actually have a CD-ROM which contains the instructions to make a human being.
Nobel Prize winner Professor Sulston oversaw the Human Genome Project, perhaps the most far-reaching scientific venture ever. At a cost of $3 billion, it drew together scientists from 18 countries across the world. Its goal? To find and record every gene in the human body. Taking samples of DNA from the average population, they looked for regular areas of activity, the genes common to all of us. Every single one was then plotted on a map. This is the human genome. After 10 years, they now know where almost all of our 35,000 genes are located. But the real task has barely begun.
Yes, I mean, what we've done with the human sequence is to read the book. We read the string of letters on the pages, okay? We haven't, or we've only just begun, to figure out which of those letters really mean anything.
Work has already begun to test each and every human gene to work out what it does. Scientists can now make an artificial gene and put it inside a cell. The model gene has a color which allows them to see what proteins it is making. Once the gene is profiled, every detail is published. All across the world, genetic labs are in a race to profile the gene combinations that make us predisposed to major illnesses. Obesity is one of the biggest health problems in America. It's estimated that a third of the nation is obese. As a health issue, it is huge, because one of the legacies of obesity is diabetes, the sixth biggest killer in the US. But while genetics companies are looking for the genes for obesity, not everyone is convinced that genes alone will ever tell us the whole story.
Almost everybody watching this program will die of a genetic disease. And by that I simply mean that things like diabetes, cancer, heart disease, all the big killers, nearly all of them have a strong inherited component. But crucially, in all those big genetic killers of the modern world, the environment is very much involved. For example, if you've got a familial tendency towards diabetes, as many, many people do, the preferred treatment is not to do gene therapy, but to stop eating sweets, to stop eating lard.
Hold your tummies in, swing your arms. Hips in, up and under.
Philadelphia has one of the highest rates of obesity in America, but it's also home to a major campaign to combat obesity and diabetes.
Swing those arms.
Gwen Foster is the city's health czar, charged with getting the city's employees into shape. For those like Gwen on the front line, the promise of a future gene test is less than helpful now.
I have a fear that if people are identified with a specific gene for a disorder, then they will say that there's no hope and just waiting for medical science to come up with the pill or the cure, which is likely not to happen in a lifetime. And the other thing, yes, is that people will feel if there is not the genetic code displayed by whatever testing, that they're scot-free. Faulty genes, I always say, may load the gun, but it's lifestyle that pulls that trigger.
While they work on their lifestyles in Philadelphia, some scientists believe that genetics may still hold the key to predicting complex conditions like diabetes. But there will need to be another phase in the genetics revolution first.
Something like obesity is a very complex set of disorders. Maybe one gene deals with 5 or 10% of the picture. There's companies like Myriad that are trying to find them and sell them one at a time. That's their business model, but it can only give a tiny piece of the picture.
Craig Venter, a pioneer of the Human Genome Project, believes genetic science has to start getting personal.
So one size doesn't fit all. While we nearly have identical physiologies, there are differences. We all differ in one out of roughly 1,200 letters of the genetic code from each other. Sometimes those differences lead obviously to some different physical characteristics, but they also lead to some different responses to the environment. In the way medicine is practiced now, we sort of deal with everything as an average.
Venter's dream is to offer us all our own genome for just $1,000. Only this, he believes, will allow us to tailor our healthcare and lifestyle effectively to the genes we individually possess.
There's subpopulations of people that if they smoke, their death rate goes up exponentially over other people that smoke, right? So if you know you're in that risk group, that extra risk group, it might give you even more motivation to change your behavior.
In his vision of the future, every child will leave the maternity ward with all his DNA stored in a CD. By the time he's an adult, he will be able to see all his genetic risks. The problem is, others will want to see them too. One of the biggest issues arising from genetic information is the health and life insurance of the future. Imagine a world a few years from now where we have all our DNA stored in a CD. By then, scientists will have worked out what every gene mutation in the human body actually does.
Who loves us today, then?
No one.
Sam's insurance company want to know if he's got a specific mutation that might make him more prone to lung cancer.
One such gene we feel may be of relevance to you, given your record as a former smoker. Oh yeah, here we go.
There are serious issues we need to deal with. We need to deal with the privacy of the information, who has access to it. We have to deal with risk of employers, governments, insurance companies potentially abusing or misusing the information.
Come here, let me have a look. Come on.
If you buy a notion that a gene will cause you to have cancer at age 35, then if you're an insurance company, you'd want to use that information. If you're an employer, you probably wouldn't want to employ the person.
You have got it. Look.
All over the world in the 21st century, the next generation is going to demand their right to privacy, their right to genetic privacy, their right not to have this information about them used as a form of discrimination or misuse or abuse of their person.
Do you know something? I can't believe this. Yesterday I was healthy and I was insurable. Today I'm healthy and I'm uninsurable.
This is a vision of a possible future. Across the world, rules vary widely on whether genetic information can be used for insurance purposes. In the UK today, companies cannot use it to set premiums because of an agreed moratorium.
We are moving steadily to a situation at the moment where there are more tests available and more people are having them. Yes, and insurers, sooner or later, when we get to that type of situation, are going to have access to some information, even if they can't rate people on it, just so that they know what the costs incurred are, so that they can protect their other policyholders and their shareholders against a potential hit.
Most insurance companies in the UK are reluctant to be the first to ask for access because of public anxiety over genetic information.
I think people are genuinely frightened about this type of information. It is new. It could be put to all different sorts of uses, not just insurance, and I think we need to respect that.
As well as discrimination, there is a deeper issue. The most fundamental information about you may actually belong to someone else.
I think the public would be surprised to know that every time a scientist or a scientific institution or life science company locates a gene, they immediately patent it. They declare it as intellectual property.
Genetic companies like Myriad have been able to patent not just the test they make, but the information itself. So that, for example, they actually own the BRCA1 gene mutation.
A lot of geneticists are very worried about the idea of patenting human genes. I share that concern. I mean, nobody's invented a human gene, they've discovered it.
I find it very difficult to understand how a company can own a gene. You know, gene belongs to the human race. I don't think they've got an argument.
You can't patent a gene itself. You can't patent something naturally occurring. That's absurd.
Breast cancer runs in my family.
My mother.
My dad's sisters.
But I found out it didn't have to be inevitable.
I found out my risk with BRCA-analysis.
Myriad are now conducting an advertising campaign to raise women's awareness of their breast cancer risks. But others see a less altruistic motive.
First of all, we need to understand this is about money as much as it's about science.
Each of Myriad's BRCA1 tests cost $1,400. Yet such tests cost many millions to develop.
I think that there's always some level of criticism that people have when commercial entities are involved. It's important to understand that these tests are currently expensive. New technologies will come to play that will actually lower the cost and make them available to a broader number of people.
A patent is given not indefinitely, but for a limited period of time to allow someone who put the money and energy into developing a particular test or a particular drug to get some benefit back, to make them take the risk. Because if I say to you, you discover this and it's in the public domain, why would you bother?
The possibility of a company profiting from these is actually a good thing, because that company is given incentive to build services, to build products that will benefit humankind. If they don't benefit humankind, nobody's going to want to pay for them. Insurance companies will not reimburse them, and people will not use them.
The driving force of genetic science today is disease prevention. This means looking at the genes you have and determining what they will do to you over the course of your life, empowering you to make decisions that can affect your chances of survival. But the ultimate in preventative medicine is to avoid damaging genes being in your body in the first place. And the only way to do that is to intercept them before you are born.
Come on in. I'll introduce you to the nice folks we've got in here today. Gwen, I'm going to start with this leg that I'm touching.
Gwen Berkowitz is having fertility treatment, but not because she is infertile. She and her husband Jeff are using a groundbreaking new procedure called pre-implantation genetic diagnosis. Their hope is that PGD will give them a child able to survive more than a few weeks. For Jeff and Gwen, there was no warning that either of them carried any kind of genetic disorder until it claimed the life of their first child 2 years ago. They have come to the sea to perform a simple ceremony to commemorate the passing of their daughter, Logan Page. Logan was born with a congenital disorder called myotonic dystrophy.
I didn't hear a cry. I heard like one little whimper, and that was it. But they brought her over to me. Like a few minutes later, they put her on my chest just for a second. I just, I was like, this is amazing. This is my baby. And it was like I was unaware of the tubes and the vent. I just felt like, this is my baby. I'm going to get to take her home in a couple days.
Before they took her to the intensive care unit, they brought me over and they let me, because they were working, they're like, there's a problem because she can't breathe on her own.
Logan never did breathe unaided. She survived just 5 weeks. Logan's genes were inherited from both her parents, but the myotonic dystrophy gene was inherited from Gwen. The affected gene is responsible for muscle control, and it goes wrong like this. As well as the normal arrangement of base letters, it has an unusual addition, a series of regular repeats.
CTG, CTG, CTG. Everyone has a certain number of these triplet 3-nucleotide repeats. Jeff happens to have 7 of them on one of his chromosomes, and on his other chromosome he has 16. And that's totally normal. Over on Gwen's side, though, she has 225 of these CGG triplets. That's too many. And that gives her some symptoms of myotonic dystrophy.
Like opening jars is when I noticed it. I can show you now with my hand. If I tighten it really tight for like 4 seconds, 5 seconds, and then try to open it, that's as fast as I can open it. So.
When Gwen's 225 repeat bases were passed on to Logan, a further mutation occurred and she inherited 1,500. The lack of muscle control in Logan's body was so severe, it was fatal. For Gwen and Jeff, there is a 50-50 chance that any future child will inherit Gwen's faulty gene. These are not good odds, and having had an affected child raises the chances of having another.
It's a lottery, and you are basically taking a chance, and you don't know how the dice are going to fall, you don't know how the cards are going to play out, and so you have to cheat.
This means using pre-implantation genetic diagnosis. In simple terms, it allows Dr. Hughes to cheat at the game of genetic cards. If every hand is an embryo, before they commit to playing that hand, Hughes can analyze it to see if it has the mutated gene. Only when he is sure it's clear will he allow the Berkowitz to play it. Gwen and Jeff have decided that their only chance of a healthy child is to use PGD. 17 of Gwen's eggs have been removed. A single sperm is injected into each egg, and they're left to grow. After 3 days, 11 eggs have successfully developed into embryos. Each is a collection of 8 cells, but enough that one can be spared. The cell they remove will be sent away to be tested for the myotonic dystrophy gene. The next day, all the biopsies arrive in Detroit at the genetics testing center run by Dr. Hughes. To find out which embryos are affected, they count the number of repeat bases they've inherited from their parents. After 8 hours of tests and analysis, Dr. Hughes has the results Gwen and Jeff have been waiting a week to receive.
Hello?
Mr. Berkowitz.
Yes.
Mark Hughes calling from Michigan.
Hi, Dr. Hughes.
Hi.
Dr. Hughes, it's Gwen.
Hi, Gwen. Well, we just got the data, and we have some wonderful news. We have results on 10 of the 11 samples. 5 of them are predicted not to have myotonic dystrophy.
The next day, Gwen and Jeff are called into the clinic for the final stage of the process, re-implanting the healthy eggs. Of the 5 clear of myotonic dystrophy, they will implant 2. The other 3 will be frozen in case the transfer doesn't succeed. The 5 embryos which tested positive will be discarded. It seems the only logical thing to do because they certainly have myotonic dystrophy. Yet it still raises an ethical problem with many people.
That's wrong, that's murder.
It's like if they had the disease and maybe that's how God wanted it to be.
To me, that raises all sorts of ethical problems. So if you have a baby that you know years from now it's going to have a problem, but does that mean you abandon the baby or abort the baby, or what do you do to, it's just kind of human beings playing God.
All we're doing is helping them start their pregnancy with a healthy child. I don't really think it plows new ethical grounds, but I'll tell you what, everybody walking down on the street has a visceral, internal, basic attitude about reproduction, and when you mix reproduction into the pot with genetics, everybody has an opinion.
In our poll, there was disagreement about whether parents should have the right to screen out embryos with hereditary diseases. Americans were the least keen, with only 2 in 5 people in favor, while twice as many Turks believe that they should.
In a society where we're all completely focused on the idea that there are standards and norms that can be engineered in advance for our children, how tolerant are we likely to be to that child who doesn't accommodate the norm, who has a disability or a handicap?
I have a daughter who has some extra material on one of her chromosomes, some abnormality. And frankly, I'm thrilled with who she is. She's not like you or me. She has delays in learning and so forth, but that's how God made her, and that's who she is.
Gwen and Jeff have had letters objecting to their decision to screen out damaged embryos.
Some of the letters that we got, not quite comparing us to Nazis, but alluding to it.
We don't want a baby with blonde hair and blue eyes, or, a girl specifically, or a boy specifically. All we want is a healthy baby, and that's it. That's all we're asking for.
Now, from the point of view of the health and happiness of the family and the child, I think that's a wonderful development. I can't see how anybody with any spark of human kindness can complain about it.
You don't have a right in this world to have a baby, all right? It's not something that... there are many people that can't have babies. There are people who have infertility. There are people that, for some reason or another, can't have their own babies. They can adopt.
In our poll, the majority of people would consider never having children if they risked passing on a serious hereditary disease. In the UK, more than 4 out of 5 would consider remaining childless. More and more people are using genetic screening to avoid passing on potentially deadly genes to their children. And this looks set to increase as the technology improves. But for some people, just screening for a condition is not enough. They need more than avoidance. They need a cure. The Nash family have pushed the technology of genetic screening to what they see as a logical next step. Using PGD, they conceived a child who was not just healthy, but could save the life of his elder sister. Molly was born with Fanconi anemia. It meant that she couldn't create her own bone marrow, a condition that would lead to leukemia and almost certain death.
They said her bone marrow would start failing about age 3. And if she wasn't transplanted by the age of 7, she would die. And Molly followed that almost. That's the one thing she's ever listened to. She followed that I mean, the day she hit 3, life started falling apart.
The only solution was a bone marrow or HLA transplant, but to work, it had to be genetically compatible. Success rates from unrelated donors were less than 20%, if you could find a match.
They also told us there's a sibling transplant, a related transplant, and the success at that point was about 80%. The one catch was that you have to have a sibling. Well, Molly's closest thing at that time to a sibling was her dog. And, you know, the chance of us having another child that had Fanconi's was 1 in 4.
Then Lisa heard about PGD and went to see one of the growing number of specialists, Dr. Charles Strom, then based in Chicago.
So I said to him, okay, so tell me this. If you can tell me if the baby has Fanconi or not, why couldn't you tell me if it has the same HLA type? And he said, I don't know that we couldn't. I've never thought of that.
So this, to me, was astonishing, and I knew immediately when I heard this proposal that this was going to revolutionize the treatment of genetic disease, and it was going to revolutionize the care of children with chronic genetic diseases.
Of 30 embryos, only 5 had the right genetic makeup. In the 4th attempt at pregnancy, the last embryo produced Adam. His bone marrow was a perfect match for Molly, by now barely healthy enough for the transplant.
And I mean, here was Molly's brother that she waited for, and here was my healthy, beautiful little boy, and here was this little tiny bag of stuff that was going to save Molly's life.
Today, both children are well and looking forward to normal, healthy lives.
I think we're very lucky that we were at a point in life where technology could help us.
When asked about situations like that facing the Nash family, the majority of people think parents should be allowed to select an embryo to help cure a sibling. Surprisingly, even in Catholic Brazil, 9 out of 10 thought the parent should have this right. But not everyone is happy with the precedent the Nashes have set. This was the first time ever that a child had been selectively conceived for any purpose other than its own health.
Now, with the new biology, we go back to conception. Wouldn't it be irresponsible of a parent not to make these changes? But then, once we begin this journey, we have to understand that this forever changes the parent-child bond. The parent becomes the architect.
People have children for all sorts of reasons. They have children to save a marriage. They have children for tax purposes. They have children accidentally. Actually, most children are conceived accidentally. And certainly, I think a reason to save the life of an older sibling is the best possible reason.
But is this the first step on the road to designer babies?
Hi, Joe. Sam.
Many scientists believe that within a few years, we will all have a personal record of our genome. And one of the most powerful uses of that technology will be for couples like Joe and Sam, who want to have children. In this ultimate future vision, they are about to select the genes they wish to pass on. In short, design their child.
Okay, first things first. A boy or a girl?
Boy. Little boy, please.
As soon as we can begin to understand what our genes tell us about who we are, well, we're going to want to make choices about our children.
Professor Greg Stock is a writer on human genetics at UCLA and sees the possibility of designer babies becoming real within the next few generations.
I think that the ability to screen for complex traits is very close, in that it is no more than a decade or so away. First, it will be people that are very wealthy and want to be sure that they don't have a child that is afflicted with various genetic diseases.
I told you about the history of haemophilia on my side of the family. I don't know if you picked it up in the database.
And then it's going to slowly move towards lesser vulnerabilities, like maybe serious depression or something of that sort, and then to choices of personality and temperament.
Why wouldn't every parent want to be able to program everything they could to make the perfect child?
Okay, then. Let's start with eyes.
Right, well, I mean, we thought my eye colour, I mean, just the eye colour, because, you know, I've got short-sightedness.
I don't know, 2020 comes as a standard.
The problem is that all of this technology, this hard path approach to using the gene, it leads to a new eugenics movement, but it's a friendly one. Don't you want a healthy baby? Don't you want to bring predictability and design principles into life so that you can have a better life and your child can have a better life?
Is there anything there that catches your eye? Sorry about the pun.
But what I'm suggesting is it could be even more devastating than the social eugenics of Hitler. Even though the intentions are honorable, the motivations are noble. Because what it does in the final analysis is reduce all of life to engineering, where our children become the ultimate shopping experience in a postmodern society.
Would you like to see him?
Yeah. Yes, please.
All right. This is the 10-week profile. What baby will look like just after birth.
No one knows if this vision will ever become reality, or how far in the future. But is it a vision most of us want to see? The idea of designer babies is universally unpopular. Even in Turkey, where the highest level of support is found, less than half are in favor.
I think that's ridiculous, trying to design a baby. Half of the pleasure of having a child is not actually knowing the sex or what the child is going to look like. You have some idea because you have the genetics of the father and the mother. So trying to pick, I think we're going off into playing God when we do that and not science.
We as human beings have always strived to do is improve ourselves. That's fine. But in terms of manipulating the DNA to give us designer babies, for me, the answer's no.
Between 80 and 90% of those polled in all countries believe gene technology should only be used to treat medical conditions.
I want a blonde baby is very different from I want a baby that's not going to die in the first 6 months of life, live on a respirator and never know the sunshine.
I think when it comes to choosing the fate of your child, I think people's and the law will very soon realize what is acceptable and what isn't.
It is now 50 years since the DNA code was broken. In that time, many lives have been transformed by genetic science. But the promise of longer, healthier living for all still seems a long way away. And the successes have raised more questions than answers. Will we overcome the technological challenges? Can we unravel the ethical dilemmas? Above all, will we learn to cope with our own expectations?
There is now this whole realm that was once beyond us that we have to make choices about, and we're going to make good choices about them and we're going to make bad choices about them. It's going to be just like anything else, and it's not going to lead us to some perfect world where there are no problems, and it's not going to lead us to destruction.
We've come an enormous way in genetics in the past 50 years. 50 years ago, it would have been utterly inconceivable to imagine that in half a century we'd have sequenced the human genome. But we have. But in science, you never know how far you've gone. You're always looking at the horizon, and the horizon recedes the further forward you go.
Do you have a name?
Adam.